The DNA is basically the building block of life and of human life, of course, also. And the DNA basically is encoding everything. So what I always tell the students is actually that you should think about this as a library. So we have about 200 different cell types, and every cell type, in principle, has the same genome. And the genome is encoded in this 4 base pairs AGTC of our DNA. And so that's the library, and now every cell needs certain books and chapters that it should read. So all the metabolic function, every cell has a Krebs cycle and does these jobs. But then there are certain books and chapters within these books that every cell has to read only if it's a neuron. So if you think about this, then actually every cell, so a neuron has the complete library, and it reads some parts of the library that any other cell also reads. But then there are specific parts of the library that only the neuron reads. Okay. The same is for the liver cells. So there are certain parts of the library that the liver cell shares with other cells, but then there are some which only the liver cell needs. And now the big question is actually, if at the beginning of development every cell has the same genome, how basically is this maintained throughout the life of the organism? And this is now when the term epigenetics, which means above the genome, comes into play. That's a term that was coined by Conrad Waddington, who was a development biologist there. And what he was trying to say is specifically this. He had this picture where the genome is a marble, and the marble can run down a valley or a hill. And there are different valleys, and it could end up in valley 1, 2, 3, or 4, and that were the different cell types. So in a way, epigenetics is everything that makes cells distinct and tells them which part of the genome they should use. Now, another example that might make this more understandable is, let's stay with the library analog. So if you have a library and it's full of books and chapters, how would a neuron now remember which chapters and which books it should read? And which has it read before, or if something happens, it should read this. So what you normally do, you put earmarks or bookmarks inside, right? And that's precisely what the epigenetics is doing. So let's say there's a learning event, and all of a sudden the neuron has to start some new gene expression programs. That means it takes a book off the shelf, reads a chapter, and now it remembers basically that this is something that I need, so it puts a bookmark in and puts it back. So these bookmarks are epigenetic modifications. And in reality, these epigenetic modifications are really placed on the DNA. So the simplest epigenetic modification is the methylation of DNA. So the cytosine can be methylated, and if it's methylated, normally that means that the gene is turned off, right? And it's a binary code almost. It's yes or no. The other key epigenetic modification that we have is the histones. So the DNA in every of our cell is about 2 meters long. So how does it fit into a nucleus, which is super tiny, right? So it's heavily coiled and it's wrapped around proteins, and these proteins are called histones. And now the idea is, if this DNA, 2 meter into 10 micrometer, is super condensed and wrapped, then it's not accessible. So the genes cannot be transcribed. Or let's stay with the library example, the books cannot be taken off the library because it's closed. Okay, and now there are mechanisms, and this is called epigenetics because let's say it's wrapped around these histones. So these histones themselves can be modified by all these modifications that we know: phosphorylation, acetylation, methylation, and so on and so forth. And there's a code that tells, for example, these genes, if, for example, histone 3, lysine 4 is methylated, then the chromatin opens up, so the library is accessible and the gene can be read. If the same location now changes and it has histone 3, not lysine 4, but lysine 27 methylated, then the whole thing closes. And what people have done now is actually that they have tried to characterize, let's say, combinatorial patterns of these histone modifications that signal to the cell this part of the genome should be accessible and this one should not be. And this one should be closed forever. This one is closed but can be activated, and things like this. And what we know is that for various reasons in all of these diseases that I talked about, these epigenetic profiles change. that could have different reasons. Basically, the enzymes do not work properly anymore, or there's just too much methyl donors, actually. So very simple approaches is to eat a methyl donor to have your epigenetic up to date. But there's also other evidence that this plays a key role in the diseases that we talked about, the brain diseases, because if you look at intellectual disability diseases and this whole neurodevelopmental diseases, where you have normally one mutation that causes then actually later on cognitive decline, then there are two groups of enzymes and proteins that are really overrepresented. The first one is, of course, everything that we know from this autism spectrum disorders, so really genes and proteins that act at the synapse. And the other one that's equally important is all these regulatory proteins that control chromatin. So I can give you a very specific example, if this is not too complicated. I told you that there's a particular pattern, so let's look at histone number 3. So there are different histones, right? 1, 2, 3, 4, and there are some other ones. And histone 3 at the position amino acid lysine 4 can be trimethylated. Then it's active. The gene is active. Now there are in the human genome and also in our nerve cells, there are 6 different enzymes that catalyze this methylation process. And there are a couple of more which also cause the demethylation. And it's always a fine balance that we know from phosphorylation. There are some enzymes that put the mark on the chromatin, and this we call writers. And there are some that remove the same mark. This we call erasers. And then there are protein complexes that read these patterns, and they are called the readers. Now, if you look at this histone 3 lysine 4, every enzyme in the human body that we know that orchestrates this process, when it's mutated, it's causing these neurodevelopmental intellectual disability disorders without any questions. So every single enzyme of the pathway, telling you that when you see now in sporadic diseases, so sporadic means we don't really know if there's a genetic cause, most likely there's one, but it's not one gene only. You also see the deregulation. So that's a super important process, right? And this is actually then something where people try to develop drugs that can reinstate this plasticity, this dynamic, this chromatin function, and then bring back the ability to learn or to recall information.
Neuroscience and beyond. Know more, get inspired.